Hello and welcome to this video laying out what embryonic research is, how it has changed over time, and what myths, misconceptions, and outright lies still exist to this day. To keep the core of this video simple, let's begin by focusing on why embryonic research is a necessary practice. The human body is complex. In order to create the intricate network of different cells, tissues, organs, systems, and ultimately a human, everything needs to be in the right place and performing the right job at the right time. For that to happen, the embryo must form the right cells at the right time, in the right way, and then discard or move those that are unneeded, unwanted, or need to be used elsewhere. This is why embryos undergo a series of stages. The first are based on simply counting the number of cells involved. It goes from one cell, or effectively a fertilized ovum, up to 16 cells, and from there it forms a blastocyst. That is a blastocyst, not a blastoise. The blastocyst will then undergo its own stages of specialization. The first is gastrulation, in which the asshole is the first thing to form. Some people do not move beyond this stage, if only in spirit. For the remainder of humanity, they form multiple layers of cells in specialized regions with special purposes. This includes the ectoderm, which becomes the skin, nervous system, hair, GI tract lining, and more. The endoderm, which becomes the remaining GI tract, respiratory system, liver, and more. And then finally the mesoderm, which makes the muscles and skeleton. From here you then get fetal development. It's roughly 4 weeks onwards. That would involve elongation of bones, muscle growth, organ development, and so on. If at any of these stages something goes wrong, you get problems. For example, if the exoderm does not form correctly, you could have a baby without a brain, although more likely a miscarriage. Without long bone development, you'd have no limbs, such as what happened with the thalidomide scandal. These are just a few, rather drastic examples. In order to either fix or stop this from happening, knowing what happens, when, where, and why is needed. If, for example, you know that congenital heart abnormalities form at a given time, you can scan babies then and there during gestation and fix it before they are born or shortly thereafter. This avoids the problem of congenital heart defects leading to infant mortality. This leads us to the early and arguably perceived as ongoing controversy. That is, how do you do this research? In the early days of embryonic research, it wasn't possible to get embryos anyway, but via donations or abortions to some extent. To be very blunt about this, the abortions were viewed as a biological hazard waste product, and as such, with appropriate approvals, licenses, and so on, they could be used for this research. You could also, under the right circumstances, use things like umbilical cord blood, given it was rich in stem cells. There are differing views on this practice, from it is as close to shedding a part of a woman's own body, to every sperm is sacred and God becomes quite irate. We won't be going into that for reasons that will become very apparent shortly. The problems with embryonic research done using, effectively, aborted fetuses is that a fetus or blastula that has been aborted is effectively an unknown variable. You have no idea of the circumstances, genetics, predispositions, conditions, and more that it either has or is inclined to. The sample itself may also have undergone all kinds of difficulties during the collection process. To be very crude about it, it's like hoping to piece together a car from car wrecks at a wrecking yard. Some might be nearly perfectly fine and are just written off by the insurer. Others might be completely destroyed with almost nothing remaining. There are also those that could have unseen damage, an unknown history, and more. The problems go on, and you won't know anything about this until you start to piece them together and they're in your hand and you're looking at them. It's sort of the same thing with aborted fetuses, and it is why aborted fetuses and similar are not really used in any kind of mainstream research. It is possible to find back-alley researchers, but they're not going to be operating legally or within confines of any respectable goals. This leads us to what is being done now, and what it is being done for. Now that we have at least somewhat adequately placed the embryonic research and what it is looking at and why, we can explore what it is and, importantly, what it is in the modern context. The what being a far more difficult question than the why of embryonic research. The first thing to note is that we have long moved on from using aborted fetuses or anything of the kind, mostly. In fact, the vast majority of embryonic research has been using stem cells or similar since 1995. Those of you not great with math, that would be nearly 30 years as of the date of this video's publication. The reason is a throwback to what we mentioned earlier. The status of aborted fetuses and embryos, along with the raft of unknown variables they carry. The stem cells allow researchers to develop a relatively isolated scenario, that is an embryo with known issues or even a clean slate within which pathologies can be induced. This is the basic necessity of research, as you must control for unavoidable variables and prevent random variables from either getting involved in your research or, worst case scenario, confounding your results. This brings us to how embryonic research is done. For the vast majority of applications, we're talking about embryonic stem cells, but there are others that we'll describe in due course. The stem cells are, let's say, ubiquitous now as a way to fix anything, but there is a role, rather more and less specific. That is, when they first appear, stem cells, depending on the stage of differentiation, can become anything. As they differentiate more, the range of things they can turn into becomes increasingly limited. For this reason, embryonic stem cells are for all intents and purposes at the very start of this chain, and therefore able to become anything. To get these stem cells, or more accurately to get human embryonic stem cells in this case, they're generally obtained from donated embryos, or they can be created using a cell from a patient and a donated egg through a process called somatic cell nuclear transfer. This effectively allows for the creation of blastocyst stages of embryos without a great deal of trouble and a lot less of the ethics issues that we've described, although there are still ethical concerns with it. The latter is the primary pathway in use today, although both are in use. It is this ability to become anything that is why stem cells are so useful in research. However, they are only so useful, and that's because they can help to understand things like how to improve IVF success rates, barriers to fertilization, issues with cell division in fertilized ovum, the appropriate location of cells once they begun dividing, and more. That is research based on a very small number of cells without a lot of roles to play just yet, effectively counting up to the 16 cells, or arguably more, but for the most part you can look at it as a very small number. To get more meaningful research results, we need to move on to the next broad stage of embryonic development, the blastula. There is also, of course, the ability to look at what stops an embryo from becoming a blastula too, so there are certain ways where this could become important, not just in terms of what is happening, but what does not happen. Assuming that you're looking at the next stage, blastula research is next, and it can be broadly split into the role of embryonic research cells that have been stimulated to divide and form a blastula. That is a spherical mass of cells that are hollow in the middle, and you can take these embryonic stem cells and stimulate them, and let them grow to this point from the initial 16 cells. That's if you want to just look at the cell growth. From here, we can then look at what the blastoid itself is doing, as that is arguably more important, and in simple terms, incredibly important. We already mentioned how at this point in embryonic development, a variety of things that are necessary for the body, the basics of the nervous system, respiratory system, and more, begin forming. This is why this research is so very important, and for the most part, it only really works up till about the 4th week, if not much less, as after the 4th week we begin looking at fetal research. In humans, the development of the blastoid is incredibly complicated. The biggest issues are going to be things like gene activation, the right movement of cells, a differentiation of cells, and so on. These same cells can also be used to create effectively more stem cells. So if the intention is to try and have effectively a model of a condition, you can develop it in these early cells, split them off and culture them and develop a new embryo from this, meaning that you arguably never need actually tap into a living being as such. You can get away without it. Once you start looking at the multiple layers of the blastoid, you then begin to look at things like the adhesion proteins that ensure the spherical shape, the layers that get formed and how they get formed, whether or not you have certain, let's say, primary cell populations, that is, the cells that are responsible for each different kind of part of the blastoid. For example, some parts of it are responsible for creating a uterine adhesion and other structures in the uterus. Others are entirely responsible for the developing embryo itself, and so you have different populations in that respect, and whether or not there are issues to do with complications of the embryo implanting successfully. Being able to adequately explore and explain what is going on using these embryonic models is arguably absolutely essential if you want to improve things like IVF or other assisted reproductive technologies. As we've already mentioned, you don't actually necessarily need to use donated embryos for this. That's because you can do it using artificially generated, effectively stem cells, although there's slightly greater complexity involved in them just being stem cells. But what you can do is grow them in microwells, and that allows you to create what is, for all intents and purposes, specific parts of the blastoid, and know what's going on using those specific cells, and allows you to look at the morphology, the genome expression, and proteomics of these cells, and even the blastocyst as a whole. If the ethics, and arguably this has been the case, of using cells that have been derived from what could have been embryos in the past is a concern, then the next option, which is arguably far less ethically fraught with issues, is to actually use somatic cells. That's effectively your skin cells, or something similar, and use either viruses, chemicals, and so on to reverse them into blastoids. This is something that has been done and can be done, and it allows for, if not necessarily the perfect model of a blastoid or blastocyst, it does allow for them to be explored to some extent. This does bring us to a kind of important, let's call it, carve-out to embryonic research, and that is international guidelines. Now, to be clear, they're guidelines, not rules, but for the most part they're fairly well followed regardless of where you are in the world, with a few sort of exceptions. Most embryonic research only runs up to 14 days. The reason for 14 days is several-fold, but the big things are the ethics of it, the idea that at 14 days you should have an idea about what's happening. At 14 days you also have a lot of differentiation of the cells, and to an extent, after 14 days you begin to look at issues around whether or not you need to look at it as an actual individual, which, yeah. Gastrulation itself is sort of along these lines, and gastrulation is an example of this, and it is a problem because it occurs at 16 days, and it's the phenomenon we mentioned earlier where you get the stratification of cells into different layers, and those different layers have very important roles to play, but they're distinct and separate roles. One of the ways we've been able to get around this is by creating 2D models using the cells, rather than using an actual embryo itself. That approach, using 2D models, was handicapped, let's say. It is, after all, effectively a single layer of cells, and you don't get a lot of depth. This has led to newer technology and research tools that allow for 3D modeling of this, but 3D modeling of gastrulation without having to actually have an embryo. Since these 3D models effectively circumvent the issue of there being an actual being, the fact is that they could never actually live outside of the, well, let's say petri dish or cell culture in which they're growing. It allowed for researchers to look at things like the primitive streak formation, neurulation, and somatogenesis. These are phenomena that if you were to try and do embryonic research and they were developing, you'd have a lot of trouble with the ethics board. Well, mostly. We'll get to those issues shortly too. Before that, we'll explain some of the ways around the issues that we've mentioned, particularly the problem of 14 days, and the idea of embryonic research going beyond that. One way has been to create culture systems that effectively make for cell cultures that are layer by layer, and artificially create an embryo, but it's not a complete embryo, it's just the necessary parts for what's being looked at. These are, for all intents and purposes, synthetic whole embryos that have been able to be cultured in the long term. One of the main advantages of these synthetic embryos is that unlike the much more narrowly tailored and defined 3D models or even 2D models, you can model a disease that will arise during the early stages of embryonic development. Congenital diseases are a good example of this. Further, due to the nature of the synthetic model, you can, to put it very crudely, poke and prod the gastroloid to achieve a very specific state of development, allowing you to not only model it, but observe what is happening, better allowing for understanding of the events that lead to things like congenital defects, failure of certain things to develop, and other embryonic growth problems. Since these are not viable organisms outside of the laboratory setting, they're not alive, although strictly speaking they're not dead either, since the cells do function to some extent. They're just not able to develop into a being. The obvious next step in this sort of research is going to be fetal research, and although this is the next step, it's also where most of the ethical and moral issues lay. Fetal models begin somewhere about 4 weeks, and this is one area where there is still scope for aborted fetuses to find a role in embryonic research, although strictly speaking it would no longer be embryonic research but fetal research. More importantly though, those sorts of fetuses wouldn't have a role to play in particular, largely down to the fact that, well, the problems we mentioned earlier, that is, so many unknown variables, and since we already have the technology and knowledge to effectively create artificial embryos, there's no reason not to continue growing them to the point where you have a fetus of a kind. The need for actual fetal samples is not that great, and it's because, well, yes, as mentioned, we have the means of culturing cells that are either from fetuses in the past, which is less used now, those that are grown from things like tissues, or the other methods mentioned. There are, let's say, 2 sources of fetal tissue that is used, although again, we are talking about a great deal of variability between labs, and different labs will have different standards. There are those that are from either elective abortions or spontaneous abortions, and there are those that are cell lines that are established by culturing fetal cells. This can be done by creating a sort of a quasi-immortal cell line, although in other cases it is just purely primary cells. It may seem rather crass to say, but the reality is that you don't need many aborted fetuses. The reason for this is very simple. The number of cells you can get and the nature of them means that they can multiply almost exponentially, which means a very small number of cells can become a huge number, which means that they, as mentioned, don't have anywhere near as much of a role in research now as they previously did. The work itself has been used in a number of ways. For example, transplantation. The reason for this is that fetal tissues lack the necessary antigens on the surface of the cells, and this means you don't get an immunological response when they are transplanted. This can be particularly important if you are trying to avoid the immune system reacting to something. The other way they get used, and this is an area of research that is much more of a problem, largely because there is an unreliable body of evidence for and against it, and it's a very, very immature topic. That is, you can transplant cells from fetuses into a mature adult body, into parts of the body where cells no longer divide or get replaced, for example, neurons or the heart. Alongside the literal fetal research, there have been successful ways to create artificial fetuses. These synthetic embryos and fetuses require no donor or parent as such. They'll often use pluripotent stem cells that they backwards engineer to be able to act the same way that a fertilized egg would. This has been made using cells from the skin and similar non-reproductive cells, hence why they need to be backwards engineered. Because these are, to some extent, not able to be long-term viable embryos and later fetuses, they're used in research because they're not going to live. In a manner of speaking, it's kind of harsh, but it also removes the ethical issues of using aborted fetuses to some extent. And this brings us to the final hurdle, that is, well, ethics. We've mentioned a lot of ethical issues so far, and embryonic research is considered fraught with ethical peril as you are destroying the developing embryo at the end. For most research, this is at or less than 14 days of development. That is the catch, most research. In the last few months and arguably years, the limits on this have been relaxed in more studies, and they have gone so far as 10 or even 14 weeks. At this point, you are well into the realm of a fetus. This is also why they are much more contentious studies. The 14-day window, depending on which kind of embryo we're talking about, is arguably perfectly fine because the cells weren't going to survive any further than that and they were never going to become a fetus. But again, this is one of those issues where individual labs and scientists are going to make decisions as to how they're going to conduct the research themselves. Some are much more conscientious than others. Some have relatively clear-cut safeguards in place and they follow those very, very carefully. Others again are more laissez-faire. To put all of this in simple terms for those who are still watching, most embryonic research is not so much different from cell-based research today. It's the same techniques, knowledge, tools, and in some cases even the same sort of cells are used, although that will vary somewhat from laboratory to laboratory. This gets more complicated when you look at the gastrula stage. This is a somewhat longer-term study and often more of a problem, as it has much more clinical significance, but also more ethical problems. The reason this is more significant is that the basics of the body form during gastrulation, so knowing where, what, why, and how is essential. We're not going to be able to do much for developing fetuses if we don't know what comes beforehand. And then we have fetal research as the final hurdle. The ethics of this are the biggest modern problem. It's also why laws exist, laws or in some cases more often guidelines, that limit the timeframes and why most institutes have very clear guidelines and rules about embryonic research. There are those who get permission to push that boundary, but they do so for a good reason. It is to understand, for example, how limbs form and how to ensure they do form, how to keep your intestines inside instead of spilling out of the fetus, how to stop major congenital defects that might kill unexpectedly, and there are more things again that get examined during these studies, but these studies are very carefully watched by most institutes. There are some countries that have more relaxed rules, but they tend to be countries with a problematic past anyway. This is why there is a gradient of both the kind of research and the results that come out. Some embryo models are really only meant to look into and in some cases only mimic specific parts of the human embryo. They are very narrowly tailored and very specific. Others look at the embryonic development and really only look at one particular pathway mechanism or similar. Others look at the whole model and how it all works together in the early developing human or human-like organism. And as such, you can't just say all embryonic research is bad. Some embryonic research is really not able to be easily differentiated by the layperson from any other cell-based research. Other research again, it most definitely can be. This overall is why embryonic research continues. It's contributing to a very important part of life, but at the same time, it's also the same part of research that tends to get a lot of flack, and not necessarily for good or bad reasons. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.